Hello everyone, welcome back to one more video session. In this session, I'll be discussing about gene interaction. Particularly, I'll be talking about a type of gene interaction which is called as incomplete dominance. It is also called as blending inheritance. Okay, so in the previous session, we have discussed about Mendel's monohybrid cross experiments. In that, he formulated two general rules, particularly the law of dominance and law of segregation. If we consider law of dominance, obviously, see, here actually the cross between homozygous tall with the homozygous dwarf plant resulted in the formation of heterozygous tall plant. That means the phenotype is similar to the homozygous tall plant, correct? But when you look into the genotype, it is entirely different. It is actually a mixture of both the male parent and female parent. You could see here, correct? Okay, so this could help you for your examination if any question related to monohybrid cross experiment, like construct a monohybrid cross experiment for axial flower position. Okay, so particularly a cross between homozygous axial flower with homozygous terminal flower. Okay, so you have to construct. So that means this is actually the English alphabets that is used to denote that particular character. So this is actually the contrasting traits of a particular character. So you can just write down actually. But one thing is clear, that according to Mendel, whatever the offspring he get in the F1 generation, which are always dominant. Okay, that is actually because of the presence of one dominant gene which is contributed by that homozygous like dominant phenotype, correct? So that is actually the thing here. Okay, so you have all heterozygous, but when you look into the phenotype, they are all dominant, correct? Okay, so suppose we consider one example. So I'm going to take the same capital T small t, because this particular alphabet or the character is familiar. Obviously, I have been discussing by taking the same example so far. So I'm not going to take any other example. I'll go with the same. Okay, I'll stick to the same actually. So this is actually our F1 due to the cross between homozygous tall and homozygous dwarf plant. Okay, so we got heterozygous tall plant. Okay, according to Mendel, he says that the characters are controlled by, what is the character here? The character is nothing but the tall character. The tall character is controlled by discrete units which are called as factors, correct? So this is a factor. So two genes, obviously, that is nothing but alleles. Okay, so which control the character that is tall. Here, what kind of interaction is happening? So that means the gene interaction. See, this is gene 1 and gene 2, but these two genes are same actually. They occur in different allelic forms. So capital T is one allele and small t is another allele of the same gene. Even also you can call it as gene 1 and gene 2. That is also there. No issues. But to avoid confusion, I say allele 1 and allele 2. So there is interaction between allele 1 and allele 2 actually. So capital T is there, that is actually the dominant gene, obviously will suppress the small t, and as a result the tall t, the capital T will express and small t get suppresses. Okay, because here capital T is stronger compared to small t, correct? So that's it. Or maybe small t is weaker compared to the capital T, the either way. Okay, so for example, now I'm taking one example. So let us assume that this is a homozygous tall plant which is crossed with homozygous dwarf plant. Okay, so here I have used 8 blocks, okay, for homozygous tall, and I have used 4 blocks for homozygous dwarf plant. Due to the cross, actually, what you expect, or according to Mendel, he should get heterozygous tall plant. That means whatever the genotype, but the F1 should be taller, or it should be tall, or it should have 8 blocks similar to the father, or similar to this homozygous tall plant, correct? 8, correct? It should not be like this. Should not have any 4 blocks. It should not be dwarf according to Mendel. What if the cross between these two produces a different phenotype? That different phenotype is the intermediate of this parent number 1 and parent number 2. See here, I have, as I said, like you used 8 blocks, 4 blocks. Because of that, I have got 6 blocked young one actually offspring. So this is 6, having 6 blocks. That means, see here, you could see the height. Okay, see, 6 blocks, correct? Okay, so that means it is the intermediate. It is not exactly similar to this parent or not exactly similar to this parent. It is a different one. So that means both the genes contributed for the character of the offspring. So this kind of gene interaction is actually called as gene interaction. So the gene is interacting. The two genes are interacting. The gene that is actually particularly the paternal gene and the maternal gene is interacted, and ultimately they both contributed. It could be, we cannot say exactly contribution or something else. I'll talk about it later. Because of that, we got intermediate character. It is a medium height plant actually, not tall, not dwarf. It is medium. Okay, so this is one example. Okay, we'll keep it. Now, let us just assume, where we take one example here. Let us just assume in this heterozygous tall plant, let us assume that capital T is compared to our Sumant. Okay, so Sumant obviously, like he is a tall fellow. Obviously, we can assume that he is stronger also. Okay, and small t is compared to Sakshat. Okay, so Sumant versus Sakshat. Okay, Sumant and Sakshat. Obviously, we know that when they are together, obviously Sumant is stronger and he will suppress Sakshat. Okay, you can also, normally go with Sumant than Sakshat because Sumant is stronger. Now, case number 1. I am talking about gene interaction. Here, what kind of gene interaction is happening? Capital T is dominating small t. So, as a result, capital T get expressed. Small t cannot get expressed. That is actually, we know it. That is according to Mendel's law of dominance. Okay, in a heterozygous condition, a gene which is dominant will dominate the recessive one. That's it. Okay, or recessive gene. Okay, and as a result, it get expressed. That is the thing. What if suddenly, Sumant loses his power or energy? Okay, so suddenly he loses his energy. So that means actually his energy level drops below the Sakshat's energy level. That means now Sakshat is stronger. See, Sakshat did not increase energy actually. It is remained as it is. But Sumant actually reduced his energy because of some reasons, and as a result, his energy level drops below Sakshat's energy level. Now who is stronger? Sakshat is stronger in this group, correct? That means Sakshat will express. But according to Mendel, the small t cannot express because it is suppressed by the capital T. It is a recessive gene. Sakshat is recessive. But no, in this case it is entirely different actually. Sakshat is expressing whereas Sumant cannot express even though he is dominant gene. Case number 1 actually. Or we'll take case number 2. What if Sakshat actually suddenly gains energy? I said in the first case where Sakshat will not gain energy, but Sumant loses his energy, it goes below Sakshat energy. As a result, Sakshat looks powerful than Sumant. What if the case number 2? Suddenly Sakshat actually increases in energy, that is 50% fold. Okay, more than 50%, than what's having right now. So the energy now doubles. Okay, so it's double now. Okay, the recessive gene is producing double enzymes or maybe proteins. Then this one, what we call the capital T or Sumant, and Sumant actually will not produce energy or loses his energy. His energy level remains as it is. Let us consider his energy level is 40, the normal energy level. But in this case, Sakshat increased his energy. That is unexpected actually. As a result, he becomes stronger. Now that is actually the case number 1. Case number 2, the Sakshat increases his energy, and as a result, Sakshat is dominant over the Sumant. That's it actually, got it. So we cannot come to the conclusion that in a heterozygous condition always T should express or small t should not express or it will be suppressed. No, it varies from species to species. As Mendel considered only Pisum sativum plant, that is pea plant, for his monohybrid as well as dihybrid or tetrahybrid crosses, even even dihybrid crosses also. Okay, he found that whenever in a heterozygous condition the dominant gene will express, but that is until 1903. Okay, so where a scientist called as the Carl Correns, this fellow, Carl Correns actually, he observed a different type of gene interaction in a plant which is called, as I'll write here, Mirabilis jalapa. Okay, it is also called as 4 o'clock plant. Okay, 4 o'clock plant. This fellow, Carl Correns, in 1903 observed in Mirabilis jalapa, that is 4 o'clock plant, that due to the cross between homozygous red colored flower plant with a homozygous white colored flower plants, okay, he got a pink in F1 generation, a heterozygous pink colored flower plant. Actually, that is entirely different. According to Mendel, in the F1 generation, the phenotype should be red, but no, he got intermediate. Very simple. So you take red color and you take white color, mix it, and what color you get? Maybe because of the power of the white color, or maybe because of the reduction in the power of the red color, the white color suppresses the red color. So as a result, white color dilutes the red color, making the phenotype the pink. That means due to the cross between homozygous red color with the homozygous recessive white color. Okay, so I'll just write white color. Okay, so as a result, he got in F1 generation pink colored flower plants. That is actually entirely against Mendel's rule, correct? According to Mendel, he says that the R should be dominant over the small r, correct? The red color, that is actually the gene responsible for the red color, should be dominant over the gene responsible for the white color. No, it didn't happen like that. It is entirely different. So I'm going to erase this and I'm going to explain incomplete dominance. It is also called as blending inheritance. Now let us just learn about the incomplete dominance. It is also called as a blending inheritance. Don't get confused with incomplete dominance and blending inheritance. Both are same. The meaning of incomplete, yes, you know it. Whereas what is the meaning of blending? Blending is nothing but mixing. Okay, that means both the genes in the F1 offspring or F1 hybrid, they interact with each other and both the characters are mixed actually. Okay, according to incomplete dominance, the definition goes like this. It is the process in which a dominant gene is incompletely dominant over the recessive gene and produces an offspring which is intermediate to their parents. That is actually incomplete dominance, or it is also blending inheritance. That means that dominant gene failed to dominate completely the recessive gene. As a result, the characters which is found in the F1 generation expressed both the parents' character. Okay, that is incomplete dominance. So here you have to remember that where the dominant gene failed to dominate completely on the recessive gene, that is actually incomplete dominance. Okay, so here as I said that Carl Correns actually conducted an experiment for the first time which actually proved Mendel's law of dominance is not exactly true. Okay, so according to him, the Carl Correns actually he conducted that experiment on a plant. Okay, so I'll write the plant name here, that is 4-o'clock plant, which is also called as Mirabilis jalapa. Please remember it, very very important. Mirabilis jalapa, it is 4-o'clock plant. Okay, and another plant I just want to write here. The second example, which is given in your NCERT. This one, Mirabilis jalapa, is not given in your NCERT. That's it, but you have to remember both. Okay, another plant, that is Antirrhinum majus. Antirrhinum majus, it is also called as snapdragon. Okay, the name of the plant is also snapdragon, or it is also called as dog flower. Okay, these are the different names for Antirrhinum majus. But Carl Correns actually conducted this incomplete dominance experiment only on 4-o'clock plant, that is Mirabilis jalapa. But the same characters were found in this another plant, that is Antirrhinum majus. There is no relation with Carl Correns and Antirrhinum, but there is a relation with Mirabilis jalapa and Carl Correns. That's what actually you have to remember. Okay, but these two different plants actually exhibited the same kind of character, that is incomplete dominance. Actually, it's a type of gene interaction. Okay, so here what we're going to do is we construct a cross, okay, a flow chart for the cross, that is incomplete dominance. First I'm going to write P1. Okay, so Carl Correns actually selected a red colored flower. Okay, so I'm using red color here. A red colored flower plants which is homozygous. So I'm going to write this as RR. So you have to write here which is homozygous red. Okay, homozygous red, which is actually which produces red colored flower. Okay, flower in a plant, and which is crossed with another homozygous white colored flower. So I'm going to write this as small r. Even I could also take like WW, but we'll go according to Mendel itself. We'll not put something which confuses us. Okay, we'll go like this itself. Okay, so a cross between homozygous red and cross between homozygous white of the same species, that is actually Mirabilis jalapa or either Anterinum. Okay, matches. Okay, whatever it is. Okay, that is P1. Let us see what is the G1. You know that during the formation of the gamete, you know, the two alleles, you know, the alleles actually they separate from each other, and each allele will enter into, so gamete actually. Okay, so each allele will enter into different gametes. That is actually according to law of segregation. So we better use red color. Okay, I'm not going to write another R, you know it. Okay, here we write small r here. This is gametes, okay, of male and female or different parents. And now I'm going to write F1. So due to the cross between R and R, or due to the fusion, so what you see is, you see a genotype which is R and this a small r, which is heterozygous. Heterozygous. Okay, you have to write like this. But the phenotype is entirely different. You know, this particular plant actually produced a pink colored flower. Okay, so that means it is the mixture of red and white color. That is the pink colored flower which were produced in the F1 generation. Okay, Carl Correns actually again now continued this experiment. Actually, you know, now I'm going with the white itself. Okay, so like this, let us not write it in red color, white itself. Okay, so now we both are actually, you know, pink colored flower. That is actually selfing, that is self pollination. As a result of self pollination, so we write P2 here, that is parental 2, and here we write G2, the gamete, the possible gamete for this. You know that there are two possible gamete, either capital R or small r, two different gametes. Okay, here also same thing. Correct. Okay, now we will write F2, and now we draw the Punnett square. Okay, and here we consider male, and here we consider female. So now I am going to write capital R, small r, the gametes of male, and here I am going to write the gametes of female, capital R, small r. So the cross, the possibilities, the probability, R into R, we get two R's. Okay, that is both capital R, capital R. Here also capital, here different, capital R, small r. Here actually capital R, small r. Here smaller, smaller. According to Mendel's law of dominance, the phenotypic ratio according to monohybrid cross experiment, simple, 3:1 actually. For 3 red color flower, we have 1 white color. But this is entirely different actually. This is incomplete dominance. Here the phenotypic ratio varies. So if we get this genotype, obviously the phenotype of that offspring will be, that is red. Correct. So I'm going to write RR is red. If we get this, obviously we already got to know that, you know, if capital R and small r, that is heterozygous condition, obviously the color will be pink. The flower's color will be pink. Okay, that is intermediate character. So I'm going to write pink, and then smaller, which is the recessive parent, that is obviously white itself. Now see here the phenotypic ratio. This is red, this is pink, this is pink, this is white. So the phenotypic ratio, I'm going to write here, the phenotypic ratio of incomplete dominance is 1:2:1. What is 1 here? 1 is red, and 2 is, that is the pink, and 1 is white. Now we'll go with the genotypic ratio. Okay, we'll write genotypic ratio here. See here, genotypic ratio. See, this is one type. This is now two types. Again, this is another type. So again, 1 capital R, and 2 capital R, small r, and is to 1 smaller. So this is now incomplete dominance. Just, you know, I'll erase this part. Okay. So, okay, fine. So now what we do is, we just, you know, write down this. This is actually the pink colored flower plants. Okay, fine. So this is actually, we got it in F1. Okay. So now what I'm going to do is, let us say that R, actually obviously it is nothing but a gene. Obviously gene means we'll draw the DNA segment. This is the gene. Now let us assume that it actually produces one enzyme. Okay, this produces one enzyme, and because of the enzyme, And substrate, I think better I will not write it, thoda confusing actually. Enzyme obviously bind to its substrate. As a result of interaction, obviously something, the color will be produced actually in the flower. Okay, so instead of that I'll just write it produces one enzyme, and this enzyme actually develops red color in that flower. Okay, the capital R. That means in the homozygous condition, both are actually having the genes, and they will help to develop the red color in the flower. That is actually thing to remember. Okay, whereas if you take smaller, I'll write here smaller, which is also having that kind of the gene where it doesn't produce any enzyme. Okay, obviously because of that, though there is no color, obviously the flower will remain white itself. Okay, so white, like that, like that we assume. So now I'm going to write, in this case, when capital R and small r actually are combined in the offspring. Okay, so this is paternal and this is maternal allele. They are combined in the offspring. So what you get is, what you expect is, there are two reasons actually. Okay, you expected red color, but instead of that we got pink color. The reason could be two reasons. One is the gene of that R, the gene of this R actually in the heterozygous condition produced enzyme which is less efficient, maybe okay, less efficient enzyme. It produced less efficient enzyme, or maybe it did not produce any enzyme or no enzyme, or maybe it could be non-functional enzyme. Okay, whatever it is, obviously, like because of this, it couldn't develop red color in the flower. Reason number one. Okay. At the same time, I'm going to take the small r here. Okay, in the same case 1 itself, where here the gene which is actually of R actually did not produce any enzyme. As a result, the flower is obviously white. Okay, that means here the enzyme which is responsible for the development of the red actually failed. Okay, failed to produce the red pigment, and as a result, that because of that, the flower will be, that is in offspring F1 generation, will be pink color. That is case number 1. Okay, or maybe I'm going to write here the case number 2. There is another possibility. What if the R actually produces normal red pigment? Because the enzymes are produced by the that gene R, and as a result, it is normal working gene. Okay. What if now small r actually produces more white pigments, or like it becomes extra stronger actually, so it produces more pigments, the white pigments. So what if like this white color diluted the red color? Okay, because they are in the same plant actually. What if that the gene, that is the enzyme which is produced from the small r, suppress the activity of the R gene where it produced the red color? What if like the white color diluted red color? There are two cases actually. That's it. Okay, so this is actually a reason. Okay, no need of it, but just I told you about it. Okay, what could be the reason of incomplete dominance? Simple. R, the gene responsible for the production of the enzyme which develops pigment in that flower, that is the red pigment, failed to express because of all these reasons. Okay, so as a result, the flower we got which is pink, or maybe like the small r in the case number 2 actually became more efficient and produced lot of white pigment, which actually diluted the red colored pigment of that capital R, which is a normal gene. Okay, so there are two postulates actually, but we are not sure about it. But one thing is sure, the pigment is developed because of the enzyme reaction, okay, between a substrate and enzyme. So that's all about it. This is called as incomplete dominance or blending inheritance. What is the meaning of blending here? So in the heterozygous condition, the capital R blends with the small r. As a result, you see a different intermediate character of their parents. So that's all about the incomplete dominance. I'm going to give you some problems based on incomplete dominance. Please solve it.